I'm in high school, so it's not good
Nine. It's all good. Gosh. I don't know. I think this is. Well, I can't even say for this project, because I haven't started since wishes for that.
Then I don't have to. Good.
She just took off from the work. And I'm thinking that.
Was it Saturday? I thought Saturday and yesterday I watched all of this topic over chapter, not she that one, or the last, the new one.
It's fun, really good. Now I'm like getting a class. That's when I'm free out of the box. So what you have to do is add like just screen to that and help change it. And then I, it's just words about like so just questions. And then it's probably just kind of like
I think that's probably studying the two last four chapters more than anything.
all those questions. It's like we don't have to do. There's some baby questions until we get them right.
Chapter 6. That's just to each one.
It's just, that's it. Smart folks, but it's also like, assignments. Easy questions. Concepts, so like they may have 100 concepts. It's sometimes like 2 or 3 solutions. And there's 2 every like 2 chapters. So other than that, it's not really hard tests. I feel like it's something I don't know, but I don't listen to any of these lectures. The lecture part, it's like Zoom that are all recording, and I don't listen to most of this because I just listen to them ramble on in class.
All right, let's get started with this now that it's 11:15. So these are the topics that we're going to start with. We're going to go over our eukaryotes and eukaryotes and our viruses. We're going to then jump into genetics, then metabolism, then immunology. And if we have time, which we probably won't because we haven't in my other classes, but if we do, then I will try to relate some of that stuff back to viruses, bacteria, fungi, whatever, to try to put them into perspective with one another. So starting off with our eukaryotes and eukaryotes and all of that stuff. With our eukaryotes. And well, first let's start off with overall all of our pathogens, all microbes and pathogens. We basically have 2 types that we would start off with. The ones that have cells and the ones that don't. We call them acellular. Ones that are acellular, these are our viruses and our prions. So, and then we have the other ones that are based on cells. So first things first, what is a cell? Right? And so the first thing that every single section that I've started this review with, you guys are the third that I'm doing. When I ask them, what is a cell? Their answer is, well, it has a nucleus. I am sorry. What? No, it doesn't. So a cell overall, the whole structure has a membrane. The whole thing has a membrane. membrane-bound unit that can self-replicate. So viruses, which we're going to review them in a little bit. Can viruses have a membrane? Yes, they can. Okay. But can they self-replicate? No. That's the issue with the viruses. Okay. Viruses aren't capable of self-replication. They have to use the host machinery and resources and stuff like that. Prions are just misfolded proteins. So do they have a membrane? No. And they also can't self-replicate in the sense that they don't make new versions of themselves. They just cause existing proteins to misfold. So they don't really like replicate so much as they just cause existing proteins to misfold. So that's their issue there. So cells are membrane-bound units that can self-replicate, whatever that means for the organism. And for our cells, we have two major types of cell-based guys. The eukaryotes, also known as prokaryotes, or the eukaryotes. So the eukaryotes, these guys are defined by the fact they have no nucleus and no true organelles. This includes bacteria and archaea. That's it for eukaryotes. For our eukaryotes, these guys do have a true nucleus. And they have true organelles, true membrane-bound organelles. An organelle is a membrane-bound structure inside of the cell with a specific function within the cell. These will include the nucleus, which its function is to house the genetic information. This includes the endoplasmic reticulum. But the endoplasmic reticulum is not as simple as you all want me to think it is. The endoplasmic reticulum has two basic forms, the rough ER and the smooth ER. What happens in the rough ER? Protein production. Why is it rough? Because ribosomes. The smooth ER is for manufacturing non-protein macromolecules, so like carbohydrates and lipids. We also have, for our organelles, we also have the Golgi. The Golgi apparatus is for packaging and shipping. In vesicles. We also have the mitochondria. Every time I ask somebody what the mitochondria does, everybody so easily and happily tells me it's the powerhouse of the cell. What does that mean? ATP production, right? That's where respiration will occur. So we know that this is where ATP is produced in the cell. This is in eukaryotes. Where does ATP get produced in bacteria? The cytoplasm is where the glycolysis and the Krebs cycle will take place. The electron transport chain always happens on a membrane because we have a set of proteins bound to a membrane where the electrons travel through it, and we call it the electron transport chain. That still exists in bacteria. So in bacteria, that happens on their inner membrane, just the inner membrane that contains their cytoplasmic membrane. Okay, so we'll talk a little bit about that too. Another thing that some eukaryotes can have. So all eukaryotes have this. Some of them might have this other structure called a chloroplast. What's that for? Right, photosynthesis. Bacteria can undergo photosynthesis too, but again, all of the proteins and molecules involved in that will all happen either in the cytoplasm or on the inner membrane of the bacteria. Okay. All right, so that is our organelles for the eukaryotes. Pick up our eukaryote groups. The ones that we talked about in class, we talked about the fungi. The fungi are basically either multicellular or single cell. They can be yeasts or molds. Those are the ones that we talked about the most, the yeasts and the molds. And if they reproduce asexually, they can produce sexually too, but we didn't talk about that. The asexual structures that we talked about in the fungi. Conidiophores and sporangiophores. I might put this on a different slide. I say different slide, different page. You know what I mean? Not really what I meant to do. Undo that. Never remember which one's the copy and which one's the duplicate. Copy. Okay. Move this onto this page. What did I do?
The circle thing.
I was trying to do that the other day. I was making notes, like going back through, like you said.
Uh-huh.
And I was like, how do I move this to a different page? And I couldn't ever figure it out.
You just have, it's just practice. Like I've gotten to using this enough that it's like a lot easier for me, but it's like, so this is the lasso tool, which is this one up in the middle, kind of up there. It looks like a lasso and it does this. And then you circle it on your GoodNotes thing and you just hold down on it and it pops up with these tools up here. And you can even like select this box here and then you can make it bigger and smaller and move it around and stuff like that. Okay, so here we are with our eukaryote groups. So the conidiophores are the ones that are open. They looked like our penicillium or our aspergillus, which kind of looked like this. It might have had little dots all around it, whatever. But the sporangiophores are covered. And so they would look more like, they might have similar structures on the inside. They've also got this membrane around the outside. So these are spore forming fungi. They're asexual spores that can like be airborne and move to other places. These are the spore forming bodies, are asexual structures, the conidiophores or the sporangiophores. Okay, moving on. Then we had, make sure I'm not missing anything. Okay, then we had a protista. This was either our algae, which are all photosynthetic. I'd like to remark that fungi are not photosynthetic ever. Right? They're either saprobes or they are chemoheterotrophs, which saprobes are a type of chemoheterotroph, but all right. But algae are photosynthetic. These are the guys that provide most of the oxygen to the earth. And then we also have the protozoa. Protozoa are always unicellular, single-celled organisms. And they are defined on their ability to be motile. And so motility types. of our protozoa. The first one will be the ones that have flagella. This would include things, or examples will be like Giardia or Trichomonas vaginalis. We also have ones that have cilia. We didn't really have any examples of that. We didn't talk about any of them that cause disease. They can, but they're fewer that have cilia. Balantidium coli is one of them, but we didn't learn about it in class, so don't worry about those. But they can have cilia to be motile, so just don't forget that can happen. For our pseudopods, pseudopodia. These are the fake feet. We also call these guys amoebas. This would include Naegleria fowleri, and acanthamoeba. Both of these guys are implicated in that meningoencephalitis. That was Pam and Gam, respectively, right? Pam and Gam. All righty. And then number 4, we have the ones that are non-motile. And these are all going to be known as apicomplexans. Examples of this one would be like malaria. It's not the only one, but it's a big one. All right. That's pretty much it for our protozoa. Oh, other things I might want to talk about with the protozoa. The trophozoite is the one that is the active one. The cyst is the one that is dormant and hardy and resistant. All right, finally, we are coming to our kingdom Animalia. And then our subkingdom is going to be the helminths. These are our parasitic worms. I'm gonna create another little page for them because my handwriting is big. So I want to make sure I cover all of this. So for our parasitic worms, our helminths, these are all multicellular. And they even have organ systems and things like that. And they even have like special life cycles that they'll go through. That's basically like going to be involved in like having to have a step in snails or something like that. Life cycles. So these are our helminths. You either are a round or you are a flatworm. Roundworms are known as cestoda. No, that's the tapeworms. I always get this part wrong. That's why I have to start off writing it down. Otherwise, I forget it. Roundworms are the nematodes. Nematoda. The flatworms are called platyhelminthes. All right. And the roundworms, the nematoda, this would include things like pinworms, or like we talked about today, Ascaris lumbricoides, the giant intestinal roundworm. Whereas the platyhelminthes can be categorized into 2 sections. This could be the tapeworms or the flukes. Tapeworms, these are known as cestota. And the flukes are known as trematoda. Examples of tapeworms would be like tinea, tholium. This is our typical tapeworm that we think of with the segments and everything like that. There's also Diphyllobothrium latum, which is the sushi tapeworm. I'm not going to write that because I'm just going to trip all over it. We already know that. So flukes, examples of our trematoda. This would be, so we've already got these guys over here. I'll give them the little dots so we can know where we are on this. For examples for our trematoda, this would be like our schistosomes, which are our liver and bladder flukes, or we typically call them our blood flukes because they are present in the blood. But we also have other flukes like Opisthothorchus sinensis and Clonorchus sinensis that are our Chinese liver flukes that block the bile duct. Okay, moving on. Genetics. So let's start off. Let's start off with replication. DNA replication, this is always making copies of DNA. Remember, our DNA nucleotides are our subunits. Nucleotides. And these will consist of a 5-carbon sugar, for DNA, it's deoxyribose. For RNA, it's ribose. So 5-carbon sugar plus a nitrogenous base. Plus phosphate. The backbone of our DNA is basically your sugar phosphate repeating. The rungs of your DNA ladder are your nitrogenous bases. DNA is what we call anti-parallel because on one strand, which you have DNA that looks like this, one backbone runs from the 5' to 3', and the other one goes the opposite direction. The backbones run in opposite direction. When we replicate DNA, it is called semi-conservative. This is because when we go from one strand to two, the daughter strand consists of one old strand that we used as a template and one new strand that was generated using that template. So we use original old strand as a template. So we go from that to 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. Where black is the old. Okay, so the new strands consist of half old and half new. That's semi-conservative. All righty. And then how actual DNA replication works. You will have a strand of DNA that gets opened up at what we call the replication fork. Right, and if this is 3 and 5, then this is 5 and 3. And as we are making our copies with our new guys, we start here. This strand is made continuously. So if this strand is continuous, and we are going, this would be the opposite of how I drew it, of course it would be. This is way too big. I always think 5' is going to start over here, and it is. It's just on the other strand. This will be 5, and this will be 3. This will be 3, and this will be 5. This will be 3. And come on, thing. When I tap you accidentally, you switch to the thing all the time. 3 and 5. Okay. So synthesis always happens in the 5' to 3' direction. So we'll write that down. All right. So here, this strand that is made continuously, which strand is that one? That's the leading strand, right? This other one, because we're still going to have to make it in the 5' to 3' direction, is made in fragments. So as this one is made in this direction, 5 to 3, and then we make this one in this direction. Five to 3. This one is made in fragments. So this one is the lagging strand. And one of those fragments called? Close enough. Okazaki. That's close enough. Okazaki fragments. All right, so here's our Okazaki fragments that are made as we're opening up that fork. We're having to still go in the 5' to 3' direction. And so we're making those in discontinuous fragments. The enzyme that actually makes the actual DNA is called DNA polymerase. And then another enzyme will come in and seal together the fragments. Does anybody remember what that enzyme is called? Ligase, exactly, DNA ligase. All right, and so this is how DNA replication works. This is just for DNA replication. Next, we're going to go into transcription. It's a different process, all right? So DNA replication, making copies of DNA, going from DNA to DNA. Next, we're going to talk about transcription. Transcription is taking your DNA template and turning it into RNA. All right. Transcription occurs in 3 steps. First one is initiation. This is where our enzyme, RNA polymerase, will bind at the starting point for this. So binds at start. For RNA polymerase, this is a specific sequence on the DNA that is called promoter. Second step is elongation, where we are going to add complementary RNA nucleotides that match the DNA sequence. So this is where we're going to compare DNA and RNA. DNA's nucleotides will be A, T, C, and G. RNA's nucleotides will be A, U, C, and G. What does A bind to? T, and T binds to A, C binds to G, and G binds to C, and same thing over here, A binds to U, U binds to A, G, C. So that U is what the difference is going to be there. DNA also has sugar called deoxyribose, whereas RNA has ribose. And DNA, we're usually in a normal healthy cell, going to see it as double-stranded, and RNA is single-stranded, typically. I didn't ever talk about the viruses. We'll come back to viruses. Talk about the genetics first. Okay, so we have this RA is known as adenine. Our T is thymine. U is uracil. C is cytosine. And G is guanine. All righty. So that's how we'll be adding those complementary nucleotides, is that we will use a DNA template strand. I don't know why that has an E in it. It is DNA template. If that DNA template was AATCTAG, then our mRNA that we make would be UUAGAUC. All right, and then our third step will be termination. We're literally going to reach a terminator sequence in the DNA that will cause it to end. The result is RNA. And we have three types of RNA. mRNA, which is our working copy of a gene. rRNA, which forms the structure of a ribosome. And then tRNA, which carries our amino acid based on the codon on the mRNA. All right, so that's transcription. Now we've made our RNA. Now we can move on to translation. Translation is where we are taking our messenger RNA and making protein from it. This also has 3 steps that are the exact same as transcription, but different things are happening in them. So our first step is initiation. The ribosome parts, called subunits, bind onto the mRNA and start always at AUG, which codes for methionine. So what's going to happen is it's going to look like this is our ribosome. These little 3 nucleotide sequences in our mRNA is called a codon. In the DNA, it's called a triplet. In the mRNA, it's called a codon. And what happens is there is a tRNA attached to an amino acid. And this thing right here, let's get different colors so we can see it better, is the anticodon. The anticodon matches up to its respective codon and carries an amino acid based on whatever that codon is. 3 nucleotides, codes for 1 amino acid. Those 3 nucleotide sequence is called a codon on the mRNA. And this is basically how initiation will start with whatever your first one is. So it's going to be AUG. Coding for methionine. And so we would have had methionine attached onto this, initially from that. And the next one will come into the next spot and attach the next amino acid. This bond that forms here is called a peptide bond. All right, and so our next step is going to be elongation. Amino acids are added according to the codon sequence. We're just going to continue to do that until we get to our termination step. When a stop codon is reached, you don't need to memorize them, but just know there are stop codons. These do not code for an amino acid, and so we call them nonsense codons. So then the ribosome dissociates. And it releases the polypeptide. All right, so that's translation. Yes.
Sorry, the mRNA, the 3 give us the codon. How do you get the anticodon?
The anticodon is on the tRNA, so that's what's going on here. This is the tRNA. And it has the anticodon on it that matches the codon. So like A to U, C to G, that stuff. So we have whatever this codon is. If this was, let's say this is A, C, then the anticodon would be U, G. and then whatever amino acids on there.
Okay.
All right, so we've got transcription and translation and replication. So I'm coming back to our organisms up here, new page, because I forgot to go over the viruses, and I don't want to leave them out. These are acellular, but we've kind of gone over genetic stuff, so at least we have a primer for that before we start talking about this. So these are acellular, these are obligate, intracellular, parasites. They must multiply inside of a host cell, period, always. And so something that all viruses have, there are only two things that all viruses must have. They must have a genome. And their genome can be DNA or RNA. And that DNA can be single-stranded or double-stranded. So can the RNA. What I meant to write. Let me make this a little bit further down. So single-stranded can be positive sense or negative sense for either of these. So suddenly we have 6 different types of genomes that viruses can potentially have, whereas cells always have DNA genomes. Viruses can have any of this BS. So cells always have double-stranded DNA genomes that can come in parts that we would call chromosomes. You might have more than one chromosome. But with viruses, their genomes can be multi-segment, they can be DNA, they can be RNA, it can be single-stranded, it can be double-stranded, and whatever. So they have to have a genome. The other thing that they have to have is a capsid. This is the protein coating around the genome. It's made of subunits called capsomeres. Some viruses can have other things. They might have an envelope. Envelope is a membrane stolen from the host cell that they wrapped themselves in during budding. Non-enveloped viruses are called naked. So some of my students made a statement previously about, oh, well, if it has an envelope, it has a membrane, those must be harder to kill. It's not true. So a lot of our cleaning agents actually affect membranes, and we've talked about that, right? So surfactants and things like that disrupt membranes. So if a virus has a membrane, it needs that membrane. It's part of how it's going to enter cells. And so if we can disrupt their membrane with our cleaning agents, then we can actually disrupt the virus's ability to like infect cells. And so an envelope having that membrane on the outside actually makes them more susceptible to our cleaning agents. It also means they're more not as resistant, they're going to dry out and things like that. So they need that membrane, and it needs to be there made of lipids, and they need to be functional lipids and stuff like that. So having an envelope actually makes them weaker, and envelope viruses are actually the weakest of all of our pathogens and are easiest to get rid of all of our pathogens. Non-enveloped naked viruses are going to be harder to get rid of, generally speaking. And so some examples of non-enveloped viruses, one example in particular would be poliovirus. Poliovirus, super tiny, it's just a capsid in a genome that causes devastating effects in people, right? Whereas when we look at an enveloped virus, COVID, flu, things like that, I tell you that those are some of the weakest, easiest to get rid of viruses, and yet look at the devastation that they cause to humanity. So don't judge a virus by its cover, right? So yes, it might be weaker and easier to get rid of if we're actually using the cleaning agents, and if you can actually interrupt them from getting spread to the other person in time. So there's that. Okay, so we have our DNA and all that. One other thing I want to mention with these guys is Let me see what, I don't want to leave anything out for you guys. Let me look at what I read about it with all this other stuff. I never talked about our bacteria. This stuff. Okay, great. So we'll come back to that, I guess, too. This is what I get for not following what I've written previously. Host cell, this is fine. Okay, so let's talk about lysogenic stuff. So lysogenic versus lytic. This is something that occurs in bacteriophages, which are viruses that infect bacteria. The lytic cycle. This is going to be when we have multiplying viruses, the multiplying phages. That burst out of the bacterial host cell. The lysogenic cycle. Again, I determined this because it has gen in it, which is for the genes. This is whenever we have the prophage genome inserts into the bacterial host genome. Basically what's happening here is that this can carry extra genes with it that give like extra traits that make it better able to survive. If the prophage brings virulence factors, so things that help it survive better, cause disease better, help it multiply, help things that help cause disease. If the prophage brings those virulence factor genes, then this causes lysogenic conversion. Again, virulence factor is a term referring to anything that helps the disease progression. All right. What I want to say about our eukaryotes, our bacteria. Come back up here to this top thing. Whoops. Bacteria, right? We don't have a nucleus. We don't have any membrane-bound organelles, right? But we do have an inner membrane always. We just call it the cytoplasmic membrane. We may have a cell wall. What are cell walls made out of in bacteria? Give you a hint, starts with a P. There we go, peptidoglycan. And we may have an outer membrane. We only see that in our gram negatives. This whole structure right here is called a cellular envelope. Not to be confused with the envelope of the viruses. All right, so our gram positives versus our gram negatives. Gram positives will be an inner membrane. Inside of that inner membrane, we'll have our DNA, we'll have ribosomes, whatever else is just floating around in the cytoplasm. There's no membrane-bound structures in there. Same thing with the gram negative, the inner membrane. We have our DNA. We have our ribosomes hanging out. All righty. On our gram positives, we will have a thick cell wall. On our gram negatives, we will have a thin cell wall. And we will have an outer membrane. And on the outer membrane of gram negatives, we will have these molecules that we will call LPS, lipopolysaccharide, or endotoxin. All right. Yeah. Okay, so now we are on to the next chapter. I say chapter, right? But our next big concepts, this was chapter 10 now. Moving on to chapter 10, we did basically our unit 1. Basic rundown of that stuff. We did our genetics, which is chapter 6. And now we're moving on to chapter 10, which is our metabolism and enzymes. So let's start with our enzymes. Enzymes are made of protein primarily, but certain kinds of enzymes called conjugated enzymes have different parts to them. The whole functional enzyme, we call it the holoenzyme. The protein portion of that, called the apoenzyme. This is where the active site is, where we bind to the substrate. Now remember what the substrate is, right? The thing that the enzyme is acting on, okay? All right, and then we have the helper molecule that we call the cofactor. The cofactor, if it is inorganic, like metallic ions, we just call it a cofactor. You might call it the inorganic cofactor, but if it's an organic molecule, it gets a special term. This will be things like vitamins or things like NAD. These are known as coenzymes. All right. When we are regulating our enzymes, you might have competitive or non-competitive inhibition. In competitive inhibition, we have the inhibitor. It binds at the active site. It literally competes with the substrate for the active site. For non-competitive inhibition, the inhibitor, anybody know where it binds? Anywhere else, yes. And so we have terms for those sites where they bind, not at the active site, it's somewhere else. These are the regulatory, or also known as the allosteric. Alrighty. And then we have our different classes of enzymes. 4 of them were way more important than the other 2. There were 6 of them. So the 4 that I'm going to remind you guys about. So we had transferases. That these move like a molecular, like they move a group to another molecule, from one molecule to another. Move groups from one molecule to another. and we had ligases. These, like DNA ligase, join smaller molecules to form larger molecules. And we had hydrolases. These are breaking down larger molecules to make smaller parts. This includes, well, I don't know how this happens, but this is stuff we talked about in chapter 10. Hydrolases, when we talk about urea hydrolysis, gelatin hydrolysis, and starch hydrolysis, the enzymes that broke urea down, which was urease, that broke starch down, which was amylase, or that broke gelatin down, which was gelatinase, those were all hydrolases. They were enzymes that broke a larger molecule down into smaller parts. In the case of amylase, we were breaking down starch into different sugar molecules, and then those sugar molecules in a cell would then be able to be used for creating ATP energy through what we're going to talk about next. I'm sure you can imagine aerobic respiration, right? Okay, so hydrolases are enzymes that break larger molecules down into smaller parts. The hydro part of it, you're probably thinking, yeah, there's water in there, it's got hydro. You're absolutely right. We also break water molecules apart and then use the H's and the OH's to kind of seal off the ends of the other molecules, the smaller parts. So we are breaking water apart. In the case of ligases, it's the exact opposite. We make water by joining things together to make the bigger molecules. Okay. All right. And our last one is our all-time favorite, oxidoreductases. These are enzymes involved in electron transfer. All right, so let's talk about redox reactions briefly. These are always going to be talking about electron transfer. One atom will gain an electron, the other atom will lose an electron. And so we have oxidation and reduction. And what is oxidation? Lose an electron. So the ways that I remember that is either Leo says GER or oil rig. So lose electron oxidized, or oxidation is loss, whatever you like with these. Okay. But that's how I break them up and how I remember which one is which. Anytime that you have chemical reactions, you almost always are going to have exchange of electrons. And so knowing what is going on there helps a lot with understanding what's going on in the chemical side of things. For our electron transfer in all of our energy production when we're creating ATP, the primary way of making ATP is through oxidative phosphorylation. It's oxidative because we've got some molecule losing an electron and some molecule gaining an electron. We've got redox reactions going there. And then that movement of electrons is basically what's going to drive us to be able to make the ATP. So ATP production driven by electron transfer. That's what we're going to be talking about next, right? To make sure I've covered everything I want to on that stuff. Metabolism. I just went right into it. I didn't even talk about, oh yeah, okay. inhibition, redox, oxidation. We did the enzymes. Okay, perfect. We're good. All right, let's go into aerobic respiration then. Don't you love it? Aerobic respiration. So there's 3 major steps in aerobic respiration: glycolysis, Krebs, and the electron transport chain. In glycolysis, the main purpose of glycolysis is to take a 6-carbon glucose and turn it into 2 3-carbon pyruvates. In the process of doing this, we will make 2 net ATP. We will also make some NADH, which is an electron carrier. So we just see it as a source of electrons. All right. Then we have an intermediate step. Where we're going to take our pyruvate and then turn it into something called acetyl-CoA. So this is a 3-carbon pyruvate and a 2-carbon acetyl-CoA. In the meantime of turning it into that, we will also produce some carbon dioxide. It's our first carbon dioxide that we're going to produce. Oh no, don't be like that. Move this down a little bit. Okay, in the Krebs cycle, we are essentially,
What's the final goal, like the end product of glycolysis?
the whole point of glycolysis is to make the pyruvate. That's literally the point. The goal of glycolysis is to make pyruvate. We do make these things also in result of glycolysis, but the whole point is to make pyruvate. And then if we get pyruvate, we can turn it into acetyl-CoA. And then in the Krebs cycle, we will take acetyl-CoA and then cycle it through the citric acid cycle and then kick off some products. We're going to make carbon dioxide. We're going to make a little bit of ATP. I think it's 2. And we're going to make a whole lot of NADH and FADH2. This is the whole point of this one. These are our electron carriers. This is the main big daddy point of the Krebs cycle, to make our electron carriers. And we're kicking off carbon dioxide in the process. All right. Then the next step is our electron transport chain. This is literally a chain of proteins. On a membrane. This is a membrane for sure. These are our phospholipids, creating a phospholipid bilayer on our membranes. Whatever, it looks totally cool and totally like a membrane. And then we have on that membrane another guy. This is ATP synthase. And these guys here, does anybody know what these proteins are called that we're going to shuttle our electrons through? They are protein, they are channels, yes. These are called cytochromes. Y'all, it's coming full circle. These are called cytochromes. So just like cytochrome C oxidase that we tested for in our aerobes, That was a cytochrome. These are cytochromes. And so this one down here at the end, guess what this one is? Cytochrome C oxidase. Okay. So I'm not going to test you that on the lecture test, but just to get you guys thinking about how these things are related, because we've been working with this stuff for long enough. Now we can try to see this full circle, right? So what happens is NADH becomes NAD, and it gives off electrons, and it gives off H pluses. Let me change some colors here. Let's see, electrons and H pluses. All right, so electrons and then H pluses. And the electrons will get shuttled through our electron transport chain, which is made up of these cytochromes. And as it is moving through all of this, good, I'm glad it did that. All right, then as it's doing that, our protons will get forced out. Why? To create this gradient of pluses. This gradient of pluses is called the proton motive force. And this is what drives the pluses to come back through here. And then we get our electrons coming off of here. So if we take an electron and we say the pluses are really H pluses. I'm just drawing them smaller so that I don't have to draw the H's a million times. So H pluses. And then in comes good old friendly old oxygen. So we'll take a half of an O2. And then this makes water. So our O2 right there, oxygen, is the final electron acceptor. In aerobic respiration. Half of oxygen. So it'll just take more electrons and more hydrogen ions. So the other half will go with other electrons and other hydrogen ions. So it'll all eventually get used up. And when it doesn't get used up properly, or if the electrons are getting mixed around, or we get too many electrons, that's when we create the toxic oxygen products that the cell has to be able to deal with catalase and superoxide dismutase and other things that are involved in helping out the toxic oxygen, reactive oxygen molecules. Okay.
So the final product of this aerobic respiration, the glycolysis, is CO2, and then the final electron transport is O2. Correct.
So glycolysis will give us pyruvate. That's really like the point of it, right? We do get some ATP and some electron carriers. Whereas in Krebs and the intermediate step, we will make the CO2 in those steps there. But the big point for that is the electron carriers. Then when we get the electron carriers, we can put them through the electron transport chain, because we might have made 2 net ATP here and maybe 2 down here in the Krebs, right? I don't want the negative, because I want you guys to think that that's negative, but we might make ATP there, we might make ATP there, but overall, so we'll make another 34 ATP down here. So net to ATP, net is about 38 ATP. So if you are a fermenter, you only go through glycolysis. And so you will make 2 ATP. You don't go on from there. So when we call something a facultative anaerobe, when oxygen is present, it will use oxygen as its final electron acceptor. It can then go through Krebs and electron transport chain. But when oxygen is not available, it'll just go through glycolysis. But the problem is that it ends up building up these electrons. In fermentation, you'll build up those electron carriers. And so they don't have anywhere to go because we can't shuttle them through the electron transport chain. Because if you shuttle them through the electron transport chain, you need oxygen to help compensate for that. So they can't do that. So what do with their electrons from their NADH? They feed it back into pyruvate and turn it into alcohol, lactic acid, and carbon dioxide. That's how they manage that. All right. So now, if we were an actual obligate anaerobe, if we're an anaerobe and we're performing anaerobic respiration, still all of these steps, not the fermentation, not facultative anaerobes, but true anaerobes, will they still go through the electron transport chain? Yes. They just won't use oxygen. Right? So they won't use oxygen. So what are they going to use? Y'all, nitrate and sulfur products. So when I tell you that an organism reduces nitrate, when I tell you that an organism reduces the sulfur and it makes H2S instead of H2O, where the F do you think that comes from? The electron transport chain, y'all. It's just using something else other than oxygen. We've been talking about it the whole damn semester. You just haven't been tying it together. So now is the time. Tie the shit together that we've been talking about. When I talk about something, nitrate is being reduced, this is where it happens. Instead of O2, we'll have nitrate. Instead of O2, we'll have some sulfur product and it'll make H2S as a result. Yeah? All right, so that's where our anaerobes will do. So if you are a facultative anaerobe, then you'll ferment. You'll just stay in glycolysis. Your cells can ferment also. If you don't get enough oxygen to your cells will ferment to try to make more ATP. It makes lactic acid as a result. The lactic acid causes inflammation in the muscle tissue because of the buildup of acid, causing issues with the pH and throwing off all of their proteins and how your cells are functioning and everything like that. And that causes that muscle soreness. Your cells ferment also. All right. So aerobic respiration. We caught up on that because now y'all know what comes next. Yes. The immunology stuff. All right. All right. I just want to make sure I'm not missing anything with this. We covered this. You guys' slide is way prettier than this one. Good. Immunology. Let's do it. Immunology. All right. So in immunology, our white blood cells, what's the other word for white blood cells? Exactly. Leukocytes. And we can split up our leukocytes into two major categories, granulocytes and agranulocytes. Literally just determined based on staining them and looking for granules, which can really spots and the dark spots staining in there. So for our granulocytes, there are kind of two groups here. The first one being the fills, and the second one being our mast cells. With the fills, didn't mean to make that one go that crazy. We have basophils, eosinophils, and neutrophils. For our agranulocytes, we have two major categories yet again, the lymphocytes and the monocytes. Monocytes are found in the blood. Lymphocytes, what are our two types of lymphocytes? T cells and B cells, right? Monocytes, if they move into the tissue, they become macrophages, which I'm going to abbreviate like this, and dendritic cells. All right, so this is the basis for all of our immunity, innate and our adaptive immunity, right? So the guys that are part of our innate stuff, these are going to be our fills and our mast cells and our monocytes and our macrophages and dendritic cells. But the monocytes, macrophages, and dendritic cells can kind of be a bridge between the two. So they're going to help activate our acquired immunity, which is going to be governed entirely by our T and our B cells. All right, so in our immune response, we have phagocytes. Our major phagocytes are going to be our neutrophils are macrophages. We'll put monocytes, actually, put monocytes. Monocytes when they're in the blood, and then they get to the tissues, they become macrophages and dendritic cells. All right, there's also some other cells that are called antigen presenting cells. That's a specific role within the immune system. Antigen presenting cells. And these antigen presenting cells are going to be our monocytes and their derivatives. As well as B cells. So I'm going to make a note here. Neutrophils are phagocytes that are not APCs. And B cells are APCs that are not phagocytes. So already you might be thinking to yourself, then how do the B cells act as antigen presenting cells? How do they get antigen to present if they are antigen presenting cells? Please don't worry, we're going to get to it. We're going to get to it, I promise. All right. So here is what is going on in our immune response. First things first, MHC. It stands for major histocompatibility complex. What does MHC do? I'm not asking you about MHC1. I'm not asking you about MHC2. Overall, what is the function of MHC? To present antigens. Exactly, to present antigen to T cells. Notice I say T cells. I did not say T helper. I did not say cytotoxic. I just said T cells. MHC, the only function it has, the only thing that it is there for is to present antigen to T cells. It doesn't present a specific antigen. It don't care what kind of antigen. It only cares in two ways because we have two different types of MHC, MHC1 and MHC2. MHC1 presents inside or endogenous antigen, whereas MHC2 presents outside or exogenous antigen, period. That's all they do, not a specific antigen, nothing. If it came from inside the cell, it's presented on MHC1. If it came from outside of the cell, it's presented on MHC2. That's all it is. It's just a molecule that presents antigen to T cells. So specifically, now we can even get a little more specific here. All nucleated cells, can become infected and house a viral infection, right? All of them can. So we'll talk about this. All nucleated cells have MHC1. And then therefore can call for help when they have an inside problem. In MHC2, only APCs have MHC2. This is, just to reiterate, just to remind you guys, our B cells, our macrophages, our dendritic cells, and our monocytes too by default because they led to our macrophages and our dendritic cell monocytes. That's it. Those are the only cells that can present outside antigen. MHC1 is only recognized by CD8, the molecule CD8. Now, we are also probably thinking, oh, CD8, cytotoxic T cells. Yes, CD8 is a molecule only found on cytotoxic T cells, so you are correct. MHC2 is only recognized by CD4. Those are our T helper cells. Come back and make sure that I am doing this justice. Yes, I'm happy with that. Okay, so now let's talk about our T cells. I'm gonna make a whole new page for it so I have the room for it. T cells. What happens is you might have, we'll say this is your antigen presenting cell. Your antigen presenting cell, this is a macrophage. It went to a site where there was an infection going on, and there's bacteria out here or viruses or whatever's outside stuff. This guy gets phagocytosed inside. Phagocytosis. And then is expressed because we got it from the outside. We put it on MHC2. And here's our antigen. Remember, MHC always presents antigen. That's all it does. We're going to present it to T cells. That's all MHC does. And so since we got this from the outside, oh my gosh, outside, MHC presents the outside antigen. MHC2 does. MHC2 presents outside antigen here. Next, this is the T cell receptor. The T cell receptor sees a specific antigen. So we know that this is a T cell. So this T cell receptor is seeing antigen presented on MHC2. What type of T cell is it? CD4 positive. It has to be because we're engaging with the MHC2. So what happens is that CD4 marker is literally interacting with the MHC2, CD4. And if it is CD4 positive, what type of T cell is it? T helper. Always. When this interaction occurs, activation signals in the form of cytokines are sent. And then that causes this guy, once it's been activated, to proliferate and differentiate. Always, we will have the T regulatory ones, T helper regulatory guys. We'll have the memory guys. Always you'll have memory and regulators, always. But then more importantly, you have TH1 and TH2 every time. Not one or the other because, oh, I'm having an inside infection, so only TH1. No, no, no. Always, no matter what, because CD4 helper cells are always activated by outside. Every time. Never are T helper cells activated from inside. Never, ever. So CD4 helper cells, they have CD4 on them. CD4 only recognizes MHC2. MHC2 only presents outside antigen, period. So your captain has to be activated from outside antigen, okay? We're following? So that T helper cell, that CD4 positive T helper cell, proliferates and differentiates, and that's where we get our T helper 1 and T helper 2 cells. So I'm going to be even more clear with this. TH1 and TH2. If this was a CD4 positive T helper cell, these guys are still CD4 positive. That doesn't change. Okay. All righty. Next step. Now you have an infected lung cell. It's infected inside. I don't know why it's doing that. With virus. Any cell has this, and it presents only inside antigen. Which MHC is this? 1. Super Bowl. Always MHC, no matter what type. MHC presents to which kind of cell? A T cell. So just start there, okay? Don't confuse yourself yet. So we know that this is going to be a T cell. And we know that this is the T cell receptor. And just like above, it sees that specific antigen. But there's a difference here. MHC1, what marker interacts with MHC1? CD8. Period. So what type of T cell is this?
That's right, cytotoxic.
Mm-hmm. All right. So that's it. And so this would be able to activate each other, again, activate with signals. But they can't do it without the go-ahead from the captain. And if this is inside stuff, that's TH1. TH1 helps mediate your MHC1 response. It's the captain that gives the go-ahead for the CD8 positive cell to activate. It can't activate on its own. It has to get the signal from the TH1 cell. But the TH1 cell got activated from outside antigen. It's the same antigen, but outside. Okay. So what does the CD4 TH2 regulate? Mm-hmm. All right. B cells that are hanging out in your lymph nodes have a receptor on their surface called IgD. This receptor will see the antigen and then bring it inside and present that same antigen, which came from outside, in MHC2. Look familiar? This has got to be a CD4 helper T cell. And then this will proliferate and differentiate into the TH2 and TH1. We already know that's both happening, so let's not forget that it's happening. But the TH2 is what's going to give the signal for activating this. Right? You following? Okay, is it starting to make a little bit more sense? I hope so. So then the B cell proliferates and differentiates into the B memory cell, as well as the plasma cell. This is our effector cell that makes antibody. What's the first antibody that it makes? IgM. Then we will class switch. And we will have Ig either G, IgA, or IgE, depending on what signals. Okay. And so IgG is the workhorse. It's in the blood. It's in the blood, and it's important for memory. It's the strongest one by far. IgA is in the mucosa. It's also known as secretory because it's secreted in the mucus. And then IgE is going to be associated with parasites and allergies. One of my students had a little way of remembering this, and they said IgA, A as in ate, like you ate something, but then I wouldn't want you guys to get confused with E being eat something, but allergies, allergy. Yeah, so that's one way that she remembered it, was A was ate, or alimentary canal, which is, you know, going to be like mucosa.
I remember like A for a chew because I sneeze a lot.
Yep, mucosa, there's that. Yep. So that is my review. Do you guys have any questions off the bat based on this? Did this at least help with the MHC stuff?
I was join it all together in one page.
Well, you can.
Because
Make your own.
it's so confusing.
Make your own.
What do you mean join it?
But you said it
Like,
goes
to
this, right?
understand it all.
Okay.
Like, cause it all goes together.
Or like everything.
Everything.
Yeah.
So I challenge you to do that for yourself.
I don't.
I'm not going to do it for you guys.
I know. I
Yeah,
wouldn't
yeah.
know where to begin though, because I don't understand it anyway.
So that's it. So start somewhere that you do understand and try to build off of one of the things that you do understand. So if you, let's say if you understand about transcription or something, then try to build off of transcription to get to this point. Because if you can't see how transcription and translation and replication relate back to this, then you're not seeing the bigger picture. Because when we proliferate those cells, what do we have to do? Replicate the DNA. We have to transcribe and make new proteins. That'll be the proteins of the new cells. Transcribe and translate. That's all happening in the background. And in order to transcribe and translate, when you're make your new DNA, all of those enzymes need energy. Where are they getting their energy from? Metabolism. And so that's why I went over these topics. I feel like these topics, like immunology, because that was the hardest one for you guys, but we got to go over the aerobic respiration and the genetics stuff, because all of that stuff is in all of this. Yes. And so I have it on here. This is my stuff, and I have it written kind of big. If you want to consolidate this down, if you already know that monocytes turn into macrophages and dendritic cells, you don't need to be rewriting that. So if that's, don't rewrite the stuff that you already know. Don't waste your time on neutrophils and the phils being, you know, granulocytes. You already know that. Focus on the stuff you don't know. We don't got a lot of time left. And so on Thursday, we will be going over some meta questions. And I say meta, I'm really going to try to make it as meta as possible. So we're going to talk about all of this stuff all at one time. And so what I'm going to do probably is ask you guys a question about, okay, a person had sexual contact with somebody who is HIV positive, and the HIV virus has gotten into their body. Now what? And we will start there, and we will go through everything we can think about in as much detail as we can. Yeah. And I hope you guys will be able to lead me for that. So be ready for that sort of discussion on Thursday. And so I think that'll start putting it into that perspective for you, Lacey, and seeing like the big picture and trying to force it all to come together in one big place. Because like when I talk to you guys about like hydrolases, I'm like, oh yeah, by the way, the hydrolysis thing, and we're like, oh yeah, we did talk about that, didn't we? And then we have talked about that previously in chapter 10, we talked about that stuff. And so also with our anaerobes and that sort of stuff. So getting into that big picture, that meta thinking, we're going to have those discussions on Thursday. So I will post this video and all of these notes on Canvas for you guys. Yeah. Huh? It'll be, I'll post it in an announcement. It'll be like an announcement to your class and they'll all be there. I'll also have the AI summary that comes along with it. I don't know if that helps anybody, but it'll be there too. So, yeah. Yeah, no problem. Whatever we can do to get you guys feeling better about this stuff.
I don't know if I'm overthinking this, the ETC, isn't that one that we have it does trap the small ones, or I think I'm taking something else, you know, when you are sieving them, it traps the smaller, is the smaller travel faster? Am I thinking of something
Yes, you're thinking
else?
of gel electrophoresis.
Okay, I'm
Yes.
like, okay.
Yes, that's gel electrophoresis. Yeah. Yeah, it's a lot.